And yeah, yes. So hello, everybody. Welcome to this Validate seminar. I have two people to introduce today. We have our main speaker, and also we have somebody who has been kindly volunteered to help out with the question and answer session. My name is Blakely Nixon. I'm the Communications and Events Coordinator at Validate. Now, our speaker is Professor Fernanda Novaes she's an assistant professor of microbiology and immunology at Ohio State. Fernanda has over 20 years experience studying the immunity to leishmaniasis in mice and humans. Her research has covered neutrophil macrophage interactions, monocyte subsets, crucial for parasite control, and she has also explored the role of CD8 cells that they play in fighting cutaneous leishmaniasis. And our other sort of contributor today is going to be Dr. Najib M. El Said, who's a professor at the University of Maryland with a PhD from Yale, and who specializes in genomics, parasitism, and host pathogen interactions. He also teaches things like bioinformatics and genomics, and he is going to be helping me curate the question and answer session, because I'm not a researcher. So I'm going to now hand over to Fernanda to begin the talk. Thank you very much for doing this. Thank you.
Thank you. All right, so thank you so much, Validate, for the invite to talk to you about the work that I've been doing in studying immunopathogenesis to cutaneous leishmaniasis. So I will go over roughly what I'll be talking about today. So initially, I'll bring up the problem, and I know most of you are familiar with leishmaniasis and cutaneous leishmaniasis, but I want to give you the perspective that I have as an immunologist and why I'm so excited and interested in studying this disease. Then I'll spend some time talking about the foundational studies that led to the work that I'm doing in my lab, and then I'll spend most of my time talking about the unpublished work. It's on bioarchives, and it's just been accepted for publication, so it should be available soon. But I'll talk most of the time about the unpublished studies on how we identify hypoxia as a major signature of the disease in leishmaniasis and how this participates in disease development. So leishmaniasis is not one disease, but a spectrum of diseases, and only some of them are represented in this slide. I don't want to discount the importance of the parasite species that you're infected with, but what I want to highlight here is how the type of immune response that you develop really determines, participates in the disease spectrum that you end up at. So you can have diffuse cutaneous leishmaniasis, and if you biopsy one of these nodules that you have hundreds and thousands of them throughout your body, you're going to have a lot of macrophages, which is the cell that the parasites primarily infect, full of parasites, and very few T cells. So at this end of the spectrum, you have uncontrolled parasite growth because you lack T cells that control the disease, and this uncontrolled parasite growth is really what is determining disease severity. And as I migrate from this end of the spectrum to this end of the spectrum, you can have individuals with localized lesions that have somewhat an efficient parasite control, but with a huge influx of T cells. So if you biopsy one of those lesions, parasites are hard to find, but what you do see is a huge influx of T cells. And parasites can metastasize from the skin to the nasopharyngeal mucosa and cause tissue damage in the mucosal tissue in mucosal disease. So you can have severe disease because you lack T cells that control the pathogen, but you can also have severe disease because you have too much inflammation caused by T cells. And in my lab, this is the end of the spectrum that I'm more interested in. We're particularly looking at cutaneous leishmaniasis that have few parasites and lots of T cells and lesions, and also we're interested in how these parasites metastasize to the nasopharyngeal mucosa. And unfortunately for those individuals, there's no human vaccine approved, and the treatment is often ineffective, particularly in the patient population that I'm working with. So in Brazil, where I have access to patient samples, the patients are treated with pentavalent antimony, which is provided by the government. And if you've heard of pentavalent antimony, you know it's a heavy metal. It's very toxic, requires 21 days of intramuscular or IV injections. And essentially, half of the patients in that population will fail the first round of treatment. And they have to go through a second and sometimes a third round of treatment. And we think this treatment is so ineffective because it targets the parasites. And these individuals, as I showed you in the previous slide, don't have as much trouble controlling the pathogen as they do controlling the immune response. So understanding the immune responses that are not involved in parasite control and are associated with pathogenesis and tissue damage has been the goal of my career in studying leishmaniasis. So how are the parasites controlled then? So I just want to bring this up. This is vector-borne disease. So when the parasites are delivered to the skin, you have dendritic cells capturing antigen, migrating to draining lymph nodes, and they produce the cytokine, a Th1 cytokine called IL-12, that's going to induce the differentiation of naive CD4 T cells into T helper 1 cells that produce a significant amount of interferon gamma, and that then activates macrophages to kill Leishmania. So we understand very well how the parasites can be controlled, so we don't want to target this pathway. So when I came from Brazil, I'm Brazilian, and I was studying leishmaniasis in Brazil, and I went to the US to try to understand what was determining disease severity. And the strategy that we used at the time, while I was at Phil Scott's lab at UPenn, was to try to understand the human disease so we could use mouse models to interrogate what was happening in disease. And the strategy that we use is first to identify what is the composition of the lesion, and that can be done by many different ways. The way we chose at the time was to perform microarray analysis, because that was a long time ago before RNA-seq was so prevalent. So we perform microarray analysis comparing healthy skin to patient biopsies with the idea that we could determine what is different between a healthy skin and a lesion with Leishmania. And then with the idea that we could use that information to generate hypotheses that we could go back to the mouse model and interrogate, is this causing disease? Is this a consequence of inflammation? And then with the idea that we could give back to the patients by proposing novel treatment strategies for therapy. So you will see me going back and forth between mouse and human work throughout this talk. So this microanalysis was a group effort with our colleagues in Brazil, and also Dan Biting at the University of Pennsylvania that was just recently hired at the time to help us at vet school to perform transcription analysis in mice and humans. And so in collaboration with Dan Biting, we obtained 10 normal skins from Corte de Pedra, which is a field site that is about a 4-hour drive from the main city where Dr. Edgar Carvalho and Lucas Carvalho have their labs in Salvador, Bahia. And we also compared those biopsies to 25 Albrusiniensis lesions. So as you can see here, this is a snapshot of a small portion of the lesion. So it doesn't recapitulate everything. We don't have the entire idea of what's happening everywhere, but it gives you a very good idea of what are the pathways that are enriched in those lesions. And then when we did this microanalysis and compare healthy skin to CL lesions, we found there were over 2,000 differentially expressed genes if you looked at CL lesions compared to healthy skin. Most of them up, but also some of them down. So trying to make sense of 2,000 differentially expressed genes is really difficult. So we performed biological pathway analysis doing gene set enrichment analysis, and then we could reduce those over 2,000 differentially expressed genes into a very small number of pathways. And that's what's shown here in the slide. So each column here is an individual that's either normal skin or Albrusiniensis lesions, and each row here is a pathway, and many pathway databases gave us a similar result, so we've combined them for simplicity. And what we found was that there are very few pathways that are present in those lesions. But one of them caught our attention very quickly, which was cytolysis. And the reason for this is when we looked at the top 10 most highly differentially expressed genes in lesions, we saw that many components of the cytotoxic granule were among those. So highlighted here in orange, you have granzyme B, for example, which is 50 times in this cohort of patients, 50 times higher in a lesion compared to healthy skin, which suggests that this pathway could be participate in disease development. So what is cytolysis? So CD8 T cells particularly, but other cells can do this as well. but today we'll focus on CD8 T cells. So CD8 T cells, once they get activated, they can produce cytokines, but one of the things that they're really, really good at is producing perforin and granzymes. And perforin and granzymes, perforin opens a pore in a target cell, and that leads to the delivery of granzymes inside a target cell, and that leads to cell death. And if you're thinking of a tumor-infected tumor cell, or if you're thinking a virally infected cell, this should be a very positive outcome, right? So you're killing the tumor, you're killing the cell where the virus needs to survive, so you can control disease by this mechanism. So it's a very, very positive mechanism in many, many situations. But then we wanted to ask whether cytolysis induced by CD8 T cells, the expression of perforin granzymes, was required for disease. And I'm going to summarize this data in one slide. So one way that we showed this was by infecting a mouse that's deficient in T cells. So we call them RAG deficient mice. They lack B and T cells. So I like this model because it's almost like an in vivo test tube. You can transfer cells that have whatever genotype you're interested in. So the first thing that we noticed when we infected this mice with Braziliensis, so this is weeks, and then this is the lesion measurement that we use. And what we saw was that when we infect reg mice, in the absence of T cells, there's no disease. You can barely see any lesion developing, but when you take the lesion out, the skin out, and you try to culture parasites, there's a ton of them. So here, essentially, you have a lot of parasites, but you don't have visual disease. But then when you transfer wild-type CD8 T cells are capable of being cytolytic. You can appreciate in this picture that these mice have this severe pathology in the skin. And but what is interesting is that these mice have the same number of parasites, which is telling us that not only CD8 T cells are not controlling the parasite, but their presence is causing tissue damage. And when we transfer CD8 T cells that are perforin deficient, well, meaning they cannot exhibit granule-mediated cytotoxicity, they're unable to produce lesions. So, and the number of parasites is the same. They're not better or less, they're not better at controlling the pathogen. They're just as bad. They just cannot mediate disease. So to summarize many years of work, my work and the work of many others, what we've identified was a pathway towards immune-mediated pathology that happens in cutaneous leishmaniasis in mice, but also in patients. What we see is that in the lesions, you have a significant amount of CD8 T cells that produce perforin and granzymes. These cells then kill target cells. And one would think that this is a positive thing. So it's very counterintuitive. You're killing the infected cells, but still you're not controlling disease. In fact, what we see is that the parasites metastasize through this mechanism to distant cutaneous sites in the mouse, and we think in humans this is associated with the development of mucosal disease. In addition, these overwhelming cell death caused by Leishmania infected cells leads to the release of damage-associated molecular patterns that then activate this pro-inflammatory pathway of the NLRP3 inflammasome that leads to the processing of this highly pro-inflammatory cytokine alpha beta, and then alpha beta induces a cascade of events that leads to chronic neutrophil recruitment to the skin. So essentially, you have CD8 T cells causing disease, and these two are the main players. And I'm going to go back to that in our recent work. But what is, besides identifying a biological pathway that's important in disease, we wanted to be able to use that information to ameliorate disease in patients. And what we found essentially was that we can block disease by blocking this pathway. So there are many drugs that we've used. Some examples are here, but there are others that people have more recently used to block this pathway of CD8 T cell mediated pathology. And some of these drugs are available for treatment in patients. Some are FDA approved, and they could be used in humans. So we can block disease by dampening granzyme and perforin expression. We can block the inflammasome. We can block IL-1, and all of this is important in blocking disease mediated by CD8 T cells. So this is an example of a treated mouse with anakinra versus an untreated mouse. So if there's any physicians in this room, they may be puzzled by how can you use drugs that are so expensive. So these are some of these drugs are extremely expensive in a neglected tropical disease. I agree with you. I wish money wasn't an issue, but I know it is. So the idea is that this is a proof of concept that there are drugs, and if you target these pathways, you can ameliorate disease. And if you think about this in the future and new drugs come around that are cheaper and they're more readily available, then these patients could benefit from it. And also, I understand that host-directed therapies, there's a lot of concern in using in infectious diseases because you always have the outcome, the concern that you're going to have outgrowth of pathogens. So the idea is to do combined therapy with antimony that targets the parasites. But also, we don't think this pathway is at all involved in the pathogen control. It's really, really mediated, immunomediated pathology. So how do we know then that the summary here is from mouse work, but how do we know that this also participates in disease in patients? So one way we showed this was to follow up that first microarray analysis that we did with an RNA-seq analysis. And what the RNA-seq analysis allowed us to do was to show variability. And we took advantage, we leveraged this variability to understand some biological things that are happening in patients. So this is work by Camila Morin that was in Phil Scott's lab as a postdoc. And as you can appreciate here, granzyme, perforin, granulysin, all components of the cytolytic granule, and also alpha beta are more expressed in patients compared to healthy skin. That's exactly what we saw in our microarray analysis. But when we look at this spread, you can see there is a fold change shown here. There's a big variability between the individuals that at the time of diagnosis, when these biopsies are taken, have this amount of alpha beta versus these individuals with this amount of alpha beta. So you have about a 200-fold change between the lowest versus the highest. So what we found, what we looked for is we followed these patients up and we asked, were they curing disease by antiparasitic drugs, or are they failing treatment at 90 days? And what we found is that those individuals that start off with these high levels of alpha beta, granulysin, and granzymes are more likely to fail antiparasitic drug treatment than those with low levels of expression of AL1, granulysin, perforin, and granzyme. So this is telling us that this pathway is not just enriched in patients, but also that it participates in their disease, right? Just like we saw in the mouse model. So I concluded this work, Camila followed up this with more patient work. But then there were so many questions when we looked at this model. A lot of it that we had to figure out this far was how CD8 T cells mediate disease, how they induce pathology, and we understood very well this step by step. But as I was starting my lab 4 years ago, I was really interested in what was really mediating this process, why CD8 T cells behave this way, and why are they pathogenic and not protective? And the reason for this to be the first question that I'd like to answer in my lab was because there's some paradox in our model. So even though I spent all this time now telling you that CD8 T cells are bad in this disease, they cause target cell death and parasite metastasis and severe pathology by neutrophil recruitment, there was evidence from other groups, but also from my own work, that CD8 T cells could have a protective phenotype. And by protective, I mean their ability to produce interferon gamma. And what trying to make sense of this paradox, what we realize is that CD8 T cell interferon gamma production was really required to push the development of T helper 1 cells to induce parasite control. And we know this happens in draining lymph nodes before the lesion even appears. So we use that information to hypothesize that CD8 T cell function depends on the tissue they're located. While CD8 T cells in draining lymph nodes are protective, CD8 T cells that are in the lesion are pathogenic. So I'm not going to talk about protection today at all. This is something that we're actively working on in the lab, but mostly what I'm going to focus on is the work that Erin Fowler in my lab has been doing since we started here at Ohio State in trying to understand why CD8 T cells become pathogenic. So our hypothesis is that there's a different expression of cytolytic markers in depending on the tissue CD8 T cells are located. So that's an easy thing to do. You can stain by flow cytometry for granzyme B in mice that are infected. And you can compare the levels of granzyme B expression in CD8 T cells that are in the adrenal lymph node or CD8 T cells that are in lesions. So that's exactly what we did. We infected mice and then we looked for granzyme B expression. So I'm going to show you a lot of flow plots like this. So this is a flow cytometry plot where we have CD8 T cells and then some other molecule, in this case granzyme B. And then I have on the right side a graph with multiple mice and showing statistics. But these are representative flow plots. So as you can appreciate this from this slide, these CD8 T cells essentially primarily express granzyme B in the skin, and they don't express granzyme B or very, very little in the adrenal lymph nodes, which suggests to us that our hypothesis might be correct, that the expression of cytolytic markers is restricted to the skin and absent in other tissues. And I must say they're parasites in both adrenal lymph nodes and lesions have parasites on them. So one possibility, there are many possibilities, right, that could explain these differences. But one possibility is that this small amount of CD8 T cells that are granzyme B positive, they quickly upregulate other chemokine receptors, for example, that would take them out of the adrenal lymph node and bring them to the lesion. So if that's true, we can't capture them because they're leaving, and then we're only seeing them once they get to the skin. So we decided to then trap them in adrenal lymph nodes using a compound called FY720. So then these CD8 T cells that are in the adrenal lymph node cannot leave. And then if there's preferential recruitment to the skin, that would mean that we would see an accumulation of granzyme B positive CD8 T cells in the adrenal lymph nodes. And that's not what we saw. Even if you block the ability of CD8 T cells to leave the adrenal lymph nodes, they can't express granzyme B, which is telling us that's not preferential recruitment, but rather they go to the lesion and they see some signal that then makes them become pathogenic. So to test this, we infected mice that we know which ones are the recipients and which ones are the donors. They express different markers. And then we purified CD8 T cells that are granzyme B negative from the adrenal lymph nodes, and then we inject them directly into the lesion. So if we just expose them to the lesion microenvironment very quickly for 2 days, will that be sufficient for the CD8 T cells to upregulate granzyme B? And that's exactly what we found. So when we transfer CD8 T cells to the lesions, they begin expressing granzyme B. If they migrate back from the lesion to the adrenal lymph nodes, they lose the ability, they don't express granzyme B, which is saying there's some signal in the skin that's driving this response. So we hypothesized that there's some signal that induces a change in CD8 T cells, and that makes them become pathogenic. So you can come up with a list of signals, or you can do transcription analysis to actually figure out what the CD8 T cells are responding to once they get to the skin. So we chose to do this unbiased approach by purifying CD8s from the adrenal lymph nodes and CD8 T cells from lesions, and then doing RNA-seq analysis. And what we saw, this is just our controls, that principal component analysis that we performed shows there is about half of the differences in that transcription analysis is with the origin where the CD8 T cells are coming from. So the CD8 T cells in the adrenal lymph nodes separate well from the CD8 T cells in lesions. And then when we looked at the genes that are most highly induced in lesions, as you can appreciate here, granzyme B is the most highly differentially expressed gene, which is telling us that our controls are working. Granzyme is really, really a good marker for the CD8 T cells in the lesions. And then when we use these differentially expressed genes to look at the pathways that are enriched in lesions, the one thing that caught our attention very quickly was a response to hypoxia. So this is a heat map of many pathways associated with hypoxia. And these are the CD8 T cells in lesions, so there are 3 replicates, and CD8 T cells in adrenal lymph nodes. And the high purple means more hypoxia. And as you can appreciate from this heat map, there is an induction. So this seems like the CD8 T cells in lesions are responding to a hypoxic microenvironment. Hypoxia is not something I knew much about or had ever worked with. So I wanted to make sure that if we're going to try to understand how hypoxia is participating in this, I wanted to make sure that this had any relevance to human disease. So then we looked in lesions in blood of patients by RNA-seq analysis. So this is what we call a hypoxic score, which is one pathway, but this is true for all the pathways that we looked at here, that there is, compared to healthy subjects in the skin, there is a significant enrichment for this hypoxic score. So the lesion seems to be more hypoxic than healthy skin. And you don't see hypoxia in the blood, which is a very good control to have, whether you're healthy or you have CL, the blood is not hypoxic. So, okay, the lesions are hypoxic in humans, or at least they have a hypoxic signature. The CD8 T cells are responding to hypoxia. So what is hypoxia and why should we care? So hypoxia is a very common microenvironment that is found in all our tissues. So when we culture our cells, frequently we culture them at 21%, which is, you know, the amount of oxygen in room air. So we're never really concerned when we're culturing our cells, exposing them to hypoxia. However, in our tissues, even the lungs have significantly less oxygen than what we do when we culture ourselves in the incubator. And particularly the skin is considered to have low oxygen. This depends on the layers, but the levels of oxygen in the skin at steady state is considered somewhat hypoxic compared to other organs. So, you know, this is a physiological thing. So the cells that are circulating in our tissues, they're always experiencing different levels of oxygen, and that can impact the way they function. And in fact, in cancer, people have been widely interested in this because hypoxia is associated with poor prognosis in patients. So tumors that are more hypoxic are frequently respond less to treatment, patients die more. And the reason for this is because there are many, many things that are changed under hypoxic conditions. The tumor cells are better in hypoxic conditions. There's a defect for the ability to die. So there's a lot that happens under hypoxia. But one of the things that is known in the literature is that hypoxia boosts CD8 T cell cytolytic activity. So you see where I'm going at here. So under low oxygen levels, if you culture CD8 T cells in hypoxic conditions in the dish with a tumor cell, they're better able to kill. They express more granzyme, they express more porphyrin, and they're more likely to kill a tumor cell. So this is telling us that maybe, you know, this hypoxic environment in the skin is triggering this granzyme B expression in those CD8 T cells. But we wanted to see first whether hypoxia was induced in Leishmania infection. So to do this, we infected mice, black 6 mice with Leishmania. And then we use this compound called pimonidazole, which is a phosphorescent, it binds to hypoxic cells covalently. So we can use an antibody against pimonidazole and you can see whether the lesion or the normal skin is hypoxic. So we looked for this hypoxic marker by confocal microscopy, but you can also do this by flow cytometry. So if you look at a naive skin and we affected the mice in the ear, so as you can appreciate, it's very thin. This in blue is DAPI, and you can see the epidermis very well. These little circles here are their hair follicles, and in the middle we have the cartilage. And as you can appreciate, there is some green staining here. There's pimonidazole, as people had seen, right? The skin is somewhat hypoxic, even in our normal skin. And particularly the hair follicles seem to be lighting up a lot for pimonidazole staining. When we infect mice, there is a huge enlargement of the skin. In this mouse here, you can see the ulcer that doesn't have any pimonidazole staining, and I'm going to go back to that later. But you do see many pockets that are intensely highlighting in green, right? That are extremely hypoxic, and then you don't see much of it in the cartilage. So this is one lesion, and this is another example that you don't have an ulcer, but you have enlargement of the epidermis, and this epidermis is really high for pimonidazole as well. So you have hypoxic pockets in the skin and also in the intact dermis. So, okay, lesions are hypoxic, and Leishmania infection induces more hypoxia in the skin. So does this impact with CD8 T cells that are recruited to the lymph node? Are they experiencing different levels of oxygen? So pimonidazole is not quantitative, but we took advantage of a phosphorescent probe that can assess the amount of oxygen in the tissue. So we infected mice and we looked at the amount of oxygen in millimeters of mercury in draining lymph nodes and the infected skin. So, and as you can appreciate here, there's significantly less oxygen that CD8 T cells are exposed to when they are in the lesion compared to draining lymph nodes. And if we convert millimeters of mercury in percent of oxygen, the CD8 T cells in the lesion is somewhat anoxic. So some of them have less than 0.5% oxygen. So it's very, very, very low in oxygen. So can we take these cells that are exposed to this X amount of oxygen and then put them in hypoxia? Will that be sufficient to change them from non-pathogenic into pathogenic? So we did this. We took CD8 T cells from draining lymph nodes and cultured them in normoxia, which is our 21% oxygen, and hypoxia. And then, as you can appreciate here, there's a significant enrichment for the expression of granzyme being the CD8 T cells, which is saying that just by exposing these cells to hypoxia, that induces the cytolytic program of CD8 T cells, which suggests to us that this is truly the signal that CD8 T cells are exposed to in those lesions that is leading to granzyme expression and pathogenesis. So this work goes very much deep into the cell, and I'm not going to show you all the data here, but what we found is that when CD8 T cells are exposed to higher levels of oxygen, they can be protective. But then as they migrate to low oxygen levels, such as the inflamed skin caused by Leishmania infection, they induce the expression of this transcription factor called LIMP1, and LIMP1 then triggers the expression of really granzyme, and then the CD8 T cells mediate pathology. So there's so many directions with which we could go with this work. And then what we decided to ask next was, what causes these lesions to become hypoxic? Why you infect with Leishmania and the level of oxygen goes down so deeply compared to a healthy skin? So we went back to our transcription analysis from patients to use that information, that variability that we see in patients, to leverage that and see if that could give us a clue of what was happening in the skin. So I showed this graph before in a different format, but as you can see, there's more hypoxia, right, or hypoxic signatures in lesions from patients compared to healthy skin. But we decided then to perform a unsupervised hierarchical clustering to ask, how can these be divided, right? And this hierarchical clustering really divided the samples, our human samples from patients, into three groups. We have those individuals that have what we call a baseline, meaning the normal levels of hypoxia in lesions, right, compared to normal skin. So this is very similar, and they kind of group together here. So in gray are healthy skin, and in purple are patients, and these are all the pathways that are associated with hypoxia that we analyzed. So you have healthy skin and human and patient skin grouping together. We call this baseline. And then when we looked at, there's a group here that has this intermediate phenotype, that it's above healthy skin, but is not as high as these individuals that we're calling hypoxic. So we essentially have three groups. So we decided to, for simplicity, I'm only going to show the baseline and hypoxic cells. So we decided to compare what was different between those individuals and these individuals by gene expression. And what we found, and we looked at this by many different formats, and I'm showing one of them, is there is a signature for granulocytes, particularly neutrophils. There's a neutrophil signature in those patients that are more hypoxic compared to those individuals that are more baseline. So the more hypoxia you have, the more neutrophils or neutrophil signature you have in these patients. So this suggests to us that neutrophils somehow are participating in this process of hypoxia. In patients, this is really hard to assess. So we then went back to the mouse model to ask how neutrophils are participating. Are they hypoxic? Are they inducing hypoxia, benefiting from hypoxia, and what's happening? So we went back to our pimonidazole staining, where we used our lesion. So this is another example of a lesion that has pimonidazole, and you can see this is a piece of tissue that is not present, but you see there are areas that are less hypoxic, and there are areas that are significantly more hypoxic based on pimidazole staining. You can appreciate this by this pixel intensity of this region here, showing that you have peaks of pimidazole, lower levels, and regions where you have high pimidazole staining. So we wanted to see how are neutrophils related to the hypoxic state of the skin. So we used pimidazole, but now by flow cytometry, and we looked right after infection, because neutrophils are recruited very early before even T cells are there. So at 2 hours and 5 hours post-infection with Leishmania, when T cells are coming in at 2 weeks, and then after healing. And if we could look at neutrophils, they never truly become hypoxic at any of those time points. But if you look at the other myeloid cells that I'm combining them here, so macrophages, monocytes, dendritic cells, all of them are hypoxic, and they kind of go down as the lesion heals. So this is suggesting to us that neutrophils might be consuming oxygen and inducing the hypoxic state of the skin. So we wanted to ask by pimidazole whether you have, neutrophil staining here, and if they're grouped in certain areas that are more or less hypoxic. And what we found is that if we color, so this Ly6G, which is a neutrophil marker, is in pink. If we look at where neutrophils are, it's essentially where hypoxia is not, and surrounding them. And you can appreciate this inverse correlation where neutrophils are present, there's very little pimidazole, and then surrounding them, you have a lot of more hypoxia, which suggesting to us that the presence of neutrophils leads to oxygen consumption, and that then triggers more hypoxia in the skin. So this is not necessarily easy to test, but one strategy that we used was to deplete neutrophils from lesions. So if we take them away, what happens to CD8 T cell responses? And that's exactly what we did. We infected mice with Leishmania, and then we took away neutrophils or gave them isotype control, so they were conventional mice. And what we found is that when we take away neutrophils, CD8 T cells are less hypoxic. So, which suggesting that neutrophils are indeed consuming oxygen and altering CD8 T cell function. And if we looked at the granzyme B expression in mice that have neutrophils and mice that don't have neutrophils, there's significantly less granzyme B being expressed in mice in the absence of neutrophils, which is telling us that neutrophils are indeed regulating CD8 T cell function and turning them from protective to pathogenic. So how are neutrophils then able to consume oxygen? There are many reasons the cell consumes oxygen, needs that to survive. But one of the things that happens when neutrophils are recruited to infection sites is that they make reactive oxygen species. So they go through a respiratory burst. And to do this, they upregulate this NADPH oxidase molecule. So we then decided to test whether if cells cannot make reactive oxygen species, and they need a lot of oxygen to do this, if they don't have reactive oxygen species, what happens then to CD8 T cells? And what we found is that indeed there's a significant reduction in the ability of CD8 T cells to produce granzyme B when mice cannot make reactive oxygen species. So to put all of this, the work that I've done before, but primarily this recent work together, what we have is this working model in which neutrophils, they're recruited very early upon Leishmania infection to produce ROS, which should control Leishmania, but in this case it doesn't. They consume a lot of oxygen, so they deplete the tissue of oxygen, leading to what we call inflammatory hypoxia. The CD8 T cells that are coming from the draining lymph nodes are recruited to the skin. They're exposed to this hypoxic environment. They express this transcription factor BLymph1 that then upregulates the expression of granzyme B. So this is great. Like we figured out again, a pathway that leads to more severe pathology caused by CD8 T cells. But then how can we use that information to benefit patients? There are some drugs that can be used to modulate hypoxia, and there's a lot of interest in biologicals for this because HIF and hypoxia is so important in tumors. However, I don't think that this is the best strategy in this case because hypoxia is not only affecting CD8 T cells, it's doing many other things to all the other immune cells, and we don't have a full understanding of the role of hypoxia in CD4 T cells and in macrophages. Have a little bit of an idea, but we don't have a full understanding of what's happening. So I don't think targeting hypoxia is the best way. But what this is telling us is that CD8 T cells are only pathogenic in the skin. You can block their ability to produce disease at the skin site, at the site of infection. So how can we use that information then to benefit patients? I was very lucky that I was contacted by Joe Trapani, which is a scientist. He's a CD8 T cell cytolytic granule biologist. And he has developed this drug, which is a porphyrin inhibitor that has been used in all the models of infection. And, he had produced a systemic treatment, and we've tried it, and it worked really well, as we would predict. If you block porphyrin, you block disease. But what Joe did when he found out about our data is that he produced then a topical version of this drug that is very stable on the high temperatures, which would be what you encounter when you go to tropical countries where Leishmania is endemic. And so he developed this cream that we could then test and see if that's sufficient to ameliorate disease in the skin. So to do this, we infected mice, and then we transferred them wild-type CD8 T cells that are going to induce pathology. And then we treated those mice when lesions develop around 3 weeks with this topical porphyrin inhibitor. And what we found was that if you look at the lesion size and also the amount of pathology that's found in those lesions, if you just apply this topical treatment to the skin and block porphyrin expression in the skin, you can block disease. And again, this is not at all involved in parasite control. The number of parasites is the same between groups because CD8 T cells really don't control Leishmania, but this is telling us that we can ameliorate disease by targeting immunopathogenic responses. And if we do this, combine it with antimony, right, that targets the parasites, we think that this could be a very good strategy to ameliorate disease. So to summarize what I've shown you today, CD8 T cells and neutrophils cooperate in promoting disease. So I began showing that CD8 T cells induce more neutrophil recruitment and chronic neutrophil recruitment. And our recent work really shows that neutrophils initiate these responses. So this is truly a feed-forward loop that leads to more severe disease in mice. And we think this is also true in patients. There are many open questions, and there are two that we're actively working on in the lab. One is, how is hypoxia then impacting the other immune cells in the tissue? As I said, we don't fully understand how, particularly CD4 T cells that are the protective cells, how they're being impacted by the hypoxic state of the skin. And if hypoxia suppresses then this protective phenotype of CD8 T cells that they exhibit in the drain lymph node. So while it's inducing pathogenesis, is it also suppressing protection? So this is something that Erin's following up with this work. So to finish, I would like to acknowledge the wonderful people in my lab that have done the work. Erin Fowler, which essentially did all the work. Klaus Mustakada did the confocal microscopy, and everybody else in the lab has been extremely helpful in Erin's work. This work would not have started if it wasn't my mentor, my PhD mentor, Camille Gianni at the Federal University of Bahia in Fiocruz, Bahia, who pushed me to come to the States and start my postdoc at the lab of Phil Scott, where all this work began, with the very close help of Tim Biting, and then Joe Trapani in Australia. I have many collaborators here at Ohio State that have been participating in this work, and then I also like to thank our financial support, and I'm happy to hear your questions. Thank you.
Hi there, I'm now just going to hand over. Thank you for your talk, Fernanda, that was brilliant. And I'm just going to hand over proceedings to Professor El Said. So thank you, Professor.
Fernanda, thank you so much for this great talk. I will lead the Q&A session with a question. And I'm sure you've thought about this. Are you considering single-cell RNA-seq analysis of the lesions, maybe to dissect the contribution of the different cell types to the pathology? And what strikes me is, I'm wondering what the proportion of CD8 T cells are in those lesions, and the granzyme expression must be off the charts to be detected when they are maybe a minority. I don't know.
So in the mouse, this is a difference. There are many differences between mice and humans, but one of the differences is the amount of CD8 T cells. So in mice, particularly SPF mice, which is what we have, right? They've never seen a pathogen. This is their first time. There are very few CD8 T cells in the skin. There are CD4s, for example. That's why we had to take advantage of certain models of transfer CD8 T cells to actually mimic the type of pathology we see in patients. Because in patients, as disease progresses, and Valderes Dutra showed this in Minas Gerais, that the ratio between CD4 to CD8 T cells changes. So you have more CD8 T cells as disease becomes more severe than CD4s. So in patients, this amount of cell death and cytolytic activity is certainly happening in much greater amounts. And we think also because other cells that are not recognizing Leishmania, that are present in patients, right? Memory cells that are recognizing all the pathogens are also recruited to the tissue. And we've shown this in mice very well, and more recently patients, that they participate in mediated disease as well. So this model really recapitulates those specific CD8 T cells. And back to your question about single-cell RNA-seq, we did perform single-cell RNA-seq, but just beginning to analyze, looking at CD4s and CD8s and looking at the hypoxic responses in those two cells and what kind of downstream effects it has in CD4s versus CD8 T cells. But people have published single-cell RNA-seq of mouse lesions, and then we're also taking advantage of that because it's publicly available. So we are looking at how this is involved as well. The other cells are involved as well.
Thank you, Fernanda. I see Madhurima has a question.
Yes, thank you for the great talk. Actually, I just wanted to know regarding, since you have showed that neutrophils that are present at the lesional sites, they have increased generation of ROS. So how do you justify that the parasites coming in contact with neutrophils, they first try to reduce the ROS levels in neutrophil for their survival? And even if the ROS is enhanced, why do you think that the increased level of the reactive oxygen species is not being able to kill the parasites there?
That's a great question. I asked this myself. So Leishmania has ways to dampen the effects of reactive oxygen species. And for some reason, also neutrophils are, at least in the major model, which is the model that I've been using, they don't control the parasite. And there is evidence, very old evidence, that they act as Trojan horses and they in fact die and release parasites and give them to macrophages, deactivate the macrophages and give them. So it may be controlling the levels of parasites somewhat, but it's not effective to the point that it really clears the disease. And in fact, in the mouse, nitric oxide is more important than reactive oxygen species in controlling Leishmania. So how this participates in humans, right? If ROS is the only way that neutrophils are doing this is unknown at the moment, but at least in this model, we don't see differences in lesion size. We don't see differences in the parasite numbers in ROS deficient or sufficient mice. And we think primarily what this is doing is changing the behavior of CD8 T cells. But that's a very good question because that's another paradox, right?
Thank you so much.
Paul, your turn.
And lovely work as always. Thanks very much. Something you didn't mention at all, I don't think, is the question of antigen specificity. And I just wonder if you could comment on whether you think cognate recognition is required for granzyme release, perforin release, and whether or not in the mouse models you would get the same effect if you used a relevant bystander T cell population, for example.
So for antigen specificity, there are a couple of things that we've done. So in this model, primarily we see the CD8 T cells interacting because we've made movies of these interactions, and primarily we see interactions between infected cells and CD8 T cells, more so than towards uninfected cells, and they kill better infected cells. It still doesn't say that this is specific because these cells that are infected may be stressed and you may have, antigen-independent, stress-dependent responses from those CD8s. So it's not clear-cut. If we've transferred CD8 T cells that cannot recognize Leishmania, they don't induce pathology. That's another way of showing this. However, this is work done by Phil Scott, by others that I participated on, and we have a recent work in patients as well, that there is a TCR independent, NKG2D dependent. So NKG2D is a receptor, that's an innate receptor that NK cells use to get activated. So there is a role for this bystander, right? CD8 T cells that are not specific to Leishmania to induce disease. And we think this is activated by cytokines such as IL-15 that activates this NKG2D expression, and then the CD8 T cells are able to kill even without seeing antigen. So in patients, you would think that this is even more relevant because people are exposed to all kinds of things throughout their lives. So they have memory cells that are circulating that could be recruited when they see inflammation, right? So we think that this is particularly important in humans.
Thanks.
No, I have a technical question. How, and a simple one really, how do you derive or measure the hypoxia score?
So we call it hypoxia score, but this is one. So it didn't have space to show all the pathways. So it's just what we're calling one of the pathways, because that was true for all the pathway databases. So they're all more hypoxic, or all of them have a greater hypoxic signature in the skin. So that's what we're calling a score, but it's really one of the pathways.
Do we have any other questions from the audience?
There's a question in the chat. Is there a different hypoxia level in different clinical forms of CL? I don't know. We haven't looked if other species of Leishmania would also cause hypoxia. I would imagine so. I think that anything that causes a severe amount of inflammation in the skin, not just Leishmania, any skin inflammatory disorder probably enhances skin hypoxia. So I would imagine that, yes, but we don't have any data relevant to this. Camila and Jenny also is asking here, so could bacteria and lesion contribute to neutrophil recruitment and hypoxia? Yes, I think so. We started doing some experiments, but we don't have any conclusive data yet because we're still establishing the model. So I only started my lab 4 years ago. It seems like a long time, but it's not, especially with the pandemic in the middle. But trying to reestablish the models that I used in Phil's lab here, it's taking longer than I would hope. But the idea is you have more bacteria, you should increase neutrophil recruitment, so that could trigger hypoxia in the skin. And there is evidence actually that some bacteria that was shown in the intestine, they can activate HIFs, which are transcription factors that are induced by hypoxia, independent of even induced hypoxia. So they produce some products, butyrate, that induces HIF expression. So I imagine that it might induce HIF expression, but also it may induce hypoxia through neutrophil recruitment.
There are no other questions. Thank you again, Fernanda, for sharing.
Oh,
Oh, there is a question.
Somebody please hear me.
Ty.
Hi, very, very interesting and comprehensive study. I was just interested in your hypothesis on the MCL lesions that are seen in patients that have lesions and eventually in this disfiguring nose, and mucosal areas, and how hypoxia may be related to it. Is it maybe also like tissue dependent? Are there more neutrophils here?
So I don't know about, this is something I've thought about, but I honestly have to say I haven't done the homework to look at what people have seen in terms of the mucosal tissue and the levels of oxygen in the mucosa and other disorders, for example. I'm not sure. It's something that I would be interested in looking at. What we think is happening is that the hypoxic state of the skin is inducing cell death, and that is leading to the recruitment to the mucosal tissue. I would imagine that this is also associated once the cells get to the mucosa and CD8 T cells are recruited, they may be participating there as well. We don't have any true evidence to suggest that. At least I don't.
Paul, you unmuted. Do you have another question, comment?
No, I'm fine, thanks.
Anyone else? In that case, Fernanda, I will thank you again for sharing your work with us
Thank you,
today.
Najib.
Fantastic work. And I hand it back to the Validate Network folks.
Hi there. No, I just want to quickly say, thank you for the amazing timing. We're literally 1 minute before the hour. So thank you very much for keeping it to time. Thank you very much for an excellent talk from Fernanda and some brilliant question sharing from Professor El Said. So I think we'll call it a day there. So thank you very much, everybody.
Thank you. Have a good one.